Hi, my name is Bryce Benoit.
And I'm Ethan McCormick.
And today we're going to be talking about Chapter 28, Immune Disorders and Antimicrobial Therapy. Here's the two subsections of the first area we're talking about, disorders and deficiencies of the immune system. Starting off strong, we have allergy, hypersensitivity, and autoimmunity. Hypersensitivity is defined as an inappropriate immune response that results in host damage. And two major types of hypersensitivity can be antibody-mediated and cell-mediated. Hypersensitivity diseases are categorized according to antigens and immune mechanisms that produce disease. They can take between minutes, such as hay fever, to hours, such as allergy to penicillin, to days, up to 2 days, poison ivy response. So the hypersensitivity, just think of it as your body overreacting to a certain immune response. Now, breaking down the types of hypersensitivity more, we have immediate hypersensitivity. This would be your allergies, antibody-mediated, and immediate hypersensitivity. This could be caused by the release of vasoactive products, which acts on blood vessels, from IgE antibody-coated mast cells. Mast cells, such as types of leukocytes, have a huge role in inflammation. It's your body's response. Reactions occur within minutes after exposure to an allergy. The antigen can be mild to life-threatening, such as anaphylaxis. And 20% of the population suffers from allergies, hay fever. I myself am not one of that 20% of the population.
Can't relate.
Next up, we have delayed type hypersensitivity, DTH. Cell-mediated hypersensitivity, T cells, response characterized by tissue damage, such as breaking out of hives, due to inflammatory responses. So the hives are a result of your body fighting back whatever response it gets. And symptoms, such as poison ivy blisters, appear several hours or even days following secondary exposure to eliciting antigens. I've had poison ivy, stays there for a bit. Typical antigens include microbes, self-antigens, and chemicals that covalently bond to the skin, creating new antigens, such as contact dermatitis. Here's a close-up picture of what we see from, this is the hives reaction. Just typical red spots, and the skin of that area will be, or the muscle of that area will be inflamed. And here we have the delayed version of poison ivy, which is characterized by more like bumps and blisters. Next up, we're moving on to autoimmune conditions. First up, what is an autoimmune disease? An autoimmune disease occurs when T and B cells are activated to produce immune reactions against self-proteins. This can result in damage to the host tissue, and some diseases are caused by autoantibodies, i.e. antibodies that react with self-antigens. Some of these are allergic encephalitis, type 1 diabetes mellitus. Here's a table of some of the autoimmune diseases we have. We have very common ones like type 1 diabetes, arthritis, hypothyroidism, male infertility, anemia, bunch of basic stuff, all affecting different areas of the body. Now, going on, we have section 2.8, which talks more about superantigens and immunodeficiencies. First off, we're starting with superantigens, which are proteins capable of eliciting a strong response because they activate more T cells than a normal immune response. They're produced by many viruses and bacteria that interact with T cell receptors, TCRs. Superantigen activated T cells may produce systematic diseases characterized by systematic inflammatory reactions. Examples: rheumatic fever, arthritis, Kowalski syndrome, dermatitis, and some forms of psoriasis.
All right.
And that's the first part of this section. Concept map. Welcome back to the second spliced together part of this study project. We'll be picking off right where we left off. We cut off with some examples, rheumatic fevers, arthritis, Kowalski syndrome, atopic dermatitis, and some forms of psoriasis. And that's all for the superantigen section. Moving on next, we have immunodeficiencies. Immunodeficiencies are active adaptive immunity, or repeating that, active adaptive immunity is critical for infectious disease resistance in animals, including humans, with deficiencies in B cells are prone to bacterial infection, while those with T cell deficiencies are prone to viral infections and cancers. So B cells lower, higher chance of bacteria. T cells lower, higher chance of viral infection and cancer. I just realized I forgot to start the timer. But we're a minute in, so we'll just go for 4. Okay. Up next, we have severe combined immune deficiency syndrome. That's a long name, so we're just going to call it SCID. Is a serious inherited congenital deficiency of both B and T cells. So both B and T cells lower, higher chance of bacterial infection and viral infection and cancer. Patients live a very restricted life, limiting their exposure to any pathogen. This is where we have immune systems that are very compromised and easily able to be attacked by viruses and bacteria. So this is where you have patients on very critical care. Next up, we have acquired immunodeficiency syndrome, aka AIDS, which is caused by HIV infections, which is the shortened version of human immunodeficiency viruses that progress and kill CD4 plus T cells. Now, these T cells that are being killed is what's stopping the body from being able to fight the viral infection that is the AIDS itself, or the HIV itself. And patients are prone to opportunistic infections and cancers since they are deficient in the T cells. So the HIV infection drops the T cell rate, which allows more cancer and more viral infections to come in. And the lack of T cells caused by that is what's commonly known as AIDS. All right, on to section 2. We have 28.3, vaccination against infectious disease, and 28.4, immunotherapy. First, we have vaccinations or immunizations. We all know what vaccines are. We've been pricked by those needles many times in our lives. Let's find out what they actually do. Deliberate exposure to an antigen which triggers an adaptive immune response intended to protect an individual against future infection by the same pathogen. So basically, we have your body being injected with an antigen which causes your body to fight it back and basically builds up a tolerance and strength so you can fight against the future pathogen whenever you actually get it in its full concentrated form. The immunogen used to induce this artificial active immunity is called a vaccine. Active immunity consists of when memory cells are generated. These memory cells are what keeps the coding that we use to fight back against the genes or the infections. In the next slide, there's only some examples for vaccines of some real major diseases: anthrax, cholera, diphtheria, and meningitis. Here we have a long list of ones, but the ones we're mainly focusing on are anthrax, cholera, diphtheria,
Flu.
meningitis, pneumonia.
Yeah.
Did they say we need the flu?
No.
No. So just these three and that one right there. But of course we have everything like, tetanus, typhoid fever, tuberculosis. Very, very inconspicuously named just plague. And paratyphoid fever, all that stuff. The nature of vaccines, which we'll get to in the next section.
All right. And this is
Concept map. Hi, welcome back to part 3 of this very fun study lesson we're doing. Picking up where we left off at the beginning of this slide right here, we have the nature of vaccines. It is recommended that infants be immunized against key infectious diseases as soon as possible so that their own active immunity can replace the temporary maternal passive immunity. So basically, whenever we're still within the womb, we receive our mother's immunity as it's passed and we are still connected. But then whenever we are severed at birth from that, we have to build up our own immunities again. It is well established that the introduction of effective vaccines into a population has reduced the incidence of formerly epidemic childhood diseases such as measles. Oops. Is that popping up? Ooh. There we go. Such as measles, mumps, and has eliminated smallpox altogether. Very cool. The nature of vaccines continue. Immunization with live cells or a virus is usually more effective than with dead or inactive material. That is why most agents used for immunization are either attenuated, weakened, or inactivated pathogens, or inactive forms of microbial products, such as toxins, aka toxoids. So, like we discussed earlier, we have weakened forms of each virus attacking our bodies so we can produce memory cells that teach us the code that we need to fight the real thing. Vaccines stop the spread of infectious diseases and death, especially in kids, and the CDC sets guidelines in the US. Immunizations usually involve a series of secondary immunizations, boosters, to produce a secondary response and a higher antibody tier. So, you gotta go in and get refreshed up basically every once in a while. Builds it up, makes it stronger. A little bit of that makes you stronger in the long run. Right here we have a photo of an infant immunization right here. And the immunization that they are receiving, usually at the young age, is we have
Mumps, measles,
influenza, meningitis, pneumonia, hepatitis, both A and B, HPV, influenza, IPV, MMR, which is the one vaccine they give for measles, mumps, and rubella, rotavirus, and varicella virus, aka chickenpox. Synthetic and genetically engineered vaccines are the product of genetically engineered antigenetic components. Conjugate vaccines, on the contrary, attach smaller antigenetic proteins to a large carrier protein to improve human immune response. Attaching a polysaccharide to a toxoid improves the immune response of the polysaccharides, usually weak. And pneumococcal vaccines are made this way. So basically, we have these vaccines that are attacking not the system as a whole, but the polysaccharides and other proteins, antigenetic proteins that we already have, and strengthening them to further strengthen us. Nucleic acid vaccines. Vaccines in which the antigens of interest are usually synthesized by the animal. DNA vaccines are bacterial plasmids that contain cloned DNA that encodes the antigen of interest. RNA vaccines combine RNA or contain bRNA sequence coding an antigen, spike protein for COVID-19 virus. Use a novel vaccine delivery method. Not injected into the animal, target proteins are cloned into the plasmid vectors and injected intramuscularly. The DNA and RNA is taken up by the host cells, and the proteins are expressed. The host then mounts an immune response to the protein. So this basically has your body change its cellular reproduction style to, in turn, produce the target protein that they're trying to fight, and then having the immune response fight that protein itself at like the DNA level. Next up, immunotherapy. Field in clinical medicine that seeks to harness and actually, we'll get to that in the next one.
Alright.
Concept map. Ain't she purty? Hi, welcome back to the fourth and final part of this adventure we're on.
Jesus Christ.
We're going to be leaving off on the last section, immunotherapy, which, as we kind of touched the surface of at the very end of the last video, is a field in clinical medicine that seeks to harness cells and other components of the immune system to fight or prevent disease, including various forms of cancer. And my stupid head forgot to start the timer, so we're just going to start it at 4:30, I think. Now, the key to immunotherapeutic approaches currently in development to combat cancer include an anti-cancer vaccination, which would, as a vaccine states, induce the cancer inside of the body at a either attenuated level or other weakened level and allow the body to fight it. And the next would be a use of checkpoint inhibitors, which would, during the reproduction stage where cancer is made and reformed, like in the body, like through cancerous growth, checkpoints that would see it, recognize the cancer more, and cause the apoptosis of the cell to prevent that. And lastly, adoptive T cell transfer. So transferring T cells from a healthier patient into the already compromised body of another patient with cancer to fight the cancer itself. Firstly, we have the anti-cancer vaccines, which can be either prophylactic, preventative, or therapeutic, treatative. Prophylactic vaccines target oncogenic, cancer-causing pathogens. So if there's no disease, then there's no cancer. Human papillomavirus, HPV, can cause cervical and other forms of cancer. Hepatitis B virus can cause liver cancer. HPV in the US, high-risk HPVs, causes 3% of all cancer in women and 2% of all cancer in men. So you have the HPV affecting more women because it targets mainly cervical, and hepatitis B can cause a liver cancer. I'd say pretty dispersed upon each gender.
Yeah, that's an STI.
Mm-hmm. Then we have
Therapeutic.
well, let's not get ahead of ourselves. Transformation in eukarya is a process by which virus infection can lead to lead cells to becoming cancerous. This is how most cancer is induced, as the cell itself mutates and then begins to transform and grow rapidly. Down here, kind of cut off, we have therapeutic anti-cancer vaccines, which are designed to stimulate the body's natural immune response to cancer. So this is ideally after cancer is already induced. And then we go into full treatment mode And I imagine anti-cancer vaccines, this would increase, T cell numbers to try and fight the cancer, I imagine. Here we have checkpoint inhibitors, whose goal is to prevent neutralization of cytotoxic T cells and enhance tumoricidal effector activity, keeping T cells going. The most successful checkpoint inhibitors are monoclonal antibodies, or MAB-based drugs. These will include pembrolizumab, marketed as Keytruda, treats metastatic melanoma and prevents skin cancer from coming back after surgery. Targets PD-1, programmed F1, or T cells, or its ligand PD-L1 on cancer cells, is solid tumors and blocks inhibition of the anti-tumor immune response and keeps cancer cells from hiding. Side effects? Autoimmune condition. Not all patients respond. Primary T cells don't respond, and secondary, new clones of tumor cells develop resistance if possible. Response linked to a specific gut microbiota. And adoptive T cell transfer, ACT, focuses on harnessing the tumor targeting ability of T cells. Two main approaches have been developed for ACT therapy. Tumor infiltrating lymphocytes, which are tumor antigen specific T cell populations that can be isolated from resected tumor tissue and cultured, activated, and amplified in vitro, then infused back into the patient, into their naturally present T cells. And then we have chimeric antigen receptors, or CARs, T cells. T cells are designed to express modified TCR protein fusion derived chimeric antigen receptors with enhanced antigen-specific targeting cancer cells. Almost there, folks. Actually, I believe this is the end. Real quick, we have the step-by-step immunotherapy of, is this anything specific? Nope. It just uses the adaptive T cell transfer, which uses the CAR stuff and boop, 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 boop.
Yep.
Binds to there, yep. Does all the fun stuff. And a section that isn't being covered, and that's it.
And then this is the final concept map.